Hi, in this short tutorial I'm going to show you how you can import your FASTQ files when you want to analyze your RNA-seq project. Now it can be bulk RNA-seq or single-cell RNA-seq, it doesn't matter. This is the way how you can import your FASTQ files to your project prior processing it. So when you create a new project, you will see this window which prompts you to select the files to import. And now you have two options. The first one is if your data is stored on your local computer, you can just drag and drop them files here in this window and add them to the project. However, if you have a backend installed and Platforma is connected to a server, you can also manipulate the files on the server. And here I'm going to click remote. So now I have the access to my remote files, which are located in this on the server I am connected to. And here I'm just going to select the folder. This is the right one. And here are some of the files that I want to process. You see, these are just like regular FASTQ files. We have, it's a paired end sequencing. It's single cell data, 10x. It has been sequenced on multiple lanes. So for each sample, you can see that we have multiple lanes, lane 1, lane 2, and for each lane there are 2 reads. So I'm just gonna select all of the files from here, click import, and you see immediately that the file names are being parsed by Platforma. So first thing, the green part is what will be assigned to each sample as a sample name. So instead of having this whole string, only this part will be assigned as a sample name by default. Now, the purple part defines the lane number. So Platforma automatically recognizes the pattern and it knows that this is the lane number and this is the number of the read. And here is the pattern which was identified automatically by default. Now, let's say I want to change something. For example, let's say I don't want to have this S1 here because we see that S1 just like is a copy over all of the files. So to remove it from the sample name, so I just want to have my sample names without this S1, I'm going to do that. I'm going to add underscore double curly brackets, star, and again double curly brackets. This means that I want to skip whatever is in between two underscores after the first one. So now the sample name will be only that part, which is what we wanted. Now I also gonna change the name for this data set, and let's name it 10x gene expression. It can be any name. This is the name of your data set, which you would refer to while processing it downstream. That is all. I'm just going to hit create. And you see that automatically the data has been added to the project. For each sample, the lanes has been recognized, and also the files with different reads. Now, one more thing which is also very important is you usually not only want to import the FASTQ files, but you also want to import some metadata. And here we have a button, metadata, and I'm going to click import metadata from a txt file. Right here, I have a TSV table. It can be CSV or TSV, it doesn't matter. The only requirement is that you have a column with the same sample name as defined right here. So other columns can be anything you want, for example, like condition or time point or tissue, anything. The only requirement is that there is a column with the sample name and the sample name matches what you defined from the pattern. So here in the import metadata, I can select one of the columns that holds the sample names. In this case, it's sample ID, and usually it's identified automatically. And we see that there have been 4 samples have been matched. And I'm going to click import. And now you see that I have added metadata value with the genotype. So we have 2 samples are wild type, 2 samples have mutation. Now this data added to the project as well, and I can click run here, and I see how these files has been added to the project. And now I can proceed and analyze these files with various softwares. For example, if we work with bulk RNA-seq data, we can use star. We have a dedicated block, star read mapping, but in this case it's a single cell data, so I'm just gonna add cell ranger block to the project. And in the next video, I'm gonna show you how to analyze your single cell data using this block.